Hi everyone, and welcome to Lab 3 for AMP in fall of 2020. This week I'm going to do two separate recordings because there are two separate parts to the lab this week. The first part is the chemistry review and molecular modeling, and the second part is pH. So what I would like to do is I'll bring up the molecular modeling lab first, and we'll go through that. And again, you're going to find both of these labs in the lab resources on Blackboard. Scroll down to Lab 3, click that open, and you'll see both the labs there that we need to complete this week. So let me bring up the chemistry review, and we'll go through that one first. Nope. Come on. Okay, here we are. Hope everyone can see this. This lab, again, review the objectives. It talks about modeling kits. Obviously, we don't have them here, but I will tell you that the black wooden balls, which represent carbon atoms, would have 4 holes in them because the carbon can make 4 bonds, and we'll go over this too. The red balls would have 2 holes because oxygen can make 2 bonds. The yellow balls would have 1 hole because hydrogen can only make 1 bond. And the blue balls, which represent nitrogen, would have 3 holes because they can make 3 bonds. Okay, so we're going through this. First of all, we need to remember what atoms are and the parts of atoms, right? So an atom consists of protons, neutrons, and electrons. The protons and the neutrons are located in the nucleus of the atom. Protons have a positive charge, neutrons have no charge, and the electrons orbit around the nucleus in defined orbitals or shells, and they have a negative charge. Atoms can form bonds between each other, as we know, and there are a couple of different types of bonds. We can talk about ionic bonds. Ionic bonds are formed between positive and negative ions, and this happens when the atom either gains or loses an electron. So clearly, if it gains an electron, it's gaining negative charge, so it's going to be a negative ion. And if it loses an electron, it's losing negative charge, so it's going to become a positive ion or cation. Okay? A covalent bond is formed when atoms share electrons, in that the electrons are going to orbit both atoms. If they are shared equally between the 2 atoms, it is a nonpolar covalent bond. There's no net charge difference. If the electrons are not shared equally, then you're going to get what's called a polar covalent bond, where you have an unequal distribution of charge. We're going to talk about that. It's particularly important when we talk about water a little bit later on. So for example, carbon, talking about atoms. But let me back up. One more kind of bond that they didn't put in here that's very important is hydrogen bond. A hydrogen bond is formed between hydrogen and another electronegative atom, because hydrogen has a plus charge. And the atoms that it's going to bind with are actually nitrogen, oxygen, and fluoride or fluorine, because those all have negative charges. Hydrogen bonds are weak bonds. They're easily formed and easily broken. So if we look at carbon, which is critically important to life, carbon has 4 electrons in its outer shell. At this point, I'm going to actually bring up I've put together a little review sheet for you on this stuff, and I want to just show you that now too, because it might make all of this a little bit more easy to understand. So here is a representation of an atom, again with the protons and neutrons in the nucleus, and the little green electrons with their negative charge orbiting in specific shells or orbitals around the nucleus. And those orbitals will contain specific numbers of electrons, which is going to give that atom certain chemical properties. So how many electrons are there per shell? The first shell, or the first orbit around the nucleus, contains 2 at a maximum. The next one out contains 8. The one out from that also contains 8. And when you have all of these shells filled with the maximum number of electrons, that atom is stable, and therefore it won't be very reactive. So if we go back now to our, let me put that down here. Back now to our lab. So again, carbon has 4 electrons in its outer shell, but it's seeking to fill the outer shell to be stable. So it would need 4 more electrons. So it can get those 4 more electrons by making 4 bonds with other atoms, and then they would each share an electron. That would give carbon its stable conformation of 8 electrons in its outer shell. And we are going to go through the practice on this together on the next page. Other macromolecules we can talk about are phospholipids and triglycerides. These contain chains of fatty acids, which are carbons and hydrogens, carbons bonded together with hydrogens around them. If those chains are all carbon-carbon single bonds, then you have hydrogens all around the carbons, we call that a saturated fat. It is saturated with the maximum number of hydrogens that you can fit around the carbon. If it contains a carbon-carbon double bond, therefore we're going to have less than the maximum number of hydrogens around the carbons, it's called an unsaturated fat or fatty acid. And if it contains multiple double bonds, then it would be a polyunsaturated fatty acid. If we look at the water molecule, and let's see a picture of a water molecule, which we have up here somewhere. There's your water molecule. So you can see that in a water molecule, we have 1 oxygen and 2 hydrogens. The electrons in a water molecule are not shared equally. The oxygen pulls the electrons closer to itself. And that's what gives water its polar property. Polar meaning it has a slightly negative charge on the oxygen side, slightly positive charge on the hydrogen side. And that's what makes water such a versatile solvent. Lots of things will react with water because water will react with anything that has a charge, either a positive charge or a negative charge. If something is not charged, if it has no net charge, it's not going to react with water. For example, fats. Fats are not charged, so therefore they do not dissolve in water. They don't react with water. And as I was saying before, here's the difference between a saturated and an unsaturated fatty acid. The saturated fatty acid has maximum number of hydrogens around those carbons because they're all single bonds between the carbons. But if we stick a double bond in there, then we're going to be short 2 hydrogens, which gives it less than the maximum number, so it is unsaturated. Now we have this little exercise here where it's asking you to go through and fill in the answers to these basic questions about atoms. We start out with hydrogen. Hydrogen has an atomic number of 1, and if we remember our periodic table, and I'm sure we don't usually remember the periodic table, if we look at this, here's a periodic table. Hydrogen has 1. If you look at H, H is for hydrogen, that's the symbol for hydrogen, and there's a 1 there, which means its atomic number, which is equivalent to the number of electrons, is 1. That's the way you read the periodic table. And then if we go back here, so hydrogen has an atomic number of 1, its symbol is H. So if it has an atomic number of 1, it means it has 1 electron, so it only has 1 electron in its outermost shell. And we know that that first shell, that first electron shell, in order to be stable, has to have 2 electrons. So is hydrogen chemically stable? No. And if it's not chemically stable, how many electrons does it need? It is seeking one more electron, right? That'll make it happy. And how many pairs of electrons would hydrogen need to share with another atom to be satisfied? One. And then it says to look at the yellow wooden ball, which is the one that represents hydrogen, and it had one hole. We can do the same with carbon. Carbon has an atomic number of 6. Its chemical symbol is C. So how many electrons are present in the outermost shell of carbon? Well, you would have 2 in the first shell, so that one would be full. Then you move to the second shell, but now you're only going to have 4 in that second shell. So is carbon chemically stable? No. How many electrons is it seeking? It's seeking 4. How many pairs of electrons would carbon need to share with another atom in order to be satisfied? 4. And if we look at that wooden ball, like we said in the beginning, it would have 4 holes. And again, we can do the same thing for oxygen. Oxygen has an atomic number of 8. Its symbol is O. So how many electrons would be present in the outermost shell? Well, you'd have 2 in the first shell, and then you would have 6 in the next shell. But that shell is not full because it can have 8. So is it chemically stable? No. So it is seeking 2 more electrons. So it would form 2 bonds. And if you look at the ball for oxygen, you would see that it would have 2 holes representing those 2 bonds. And then lastly, if we look at nitrogen, atomic number 7, its symbol is N. How many electrons would be in the outermost shell? Well, again, we'll have 2 in the first. That's only going to leave 5 in that second shell. So it would need to gain 3 more electrons. So it could form 3 bonds. And if we look at that ball, we would see that there are 3 holes in it. These next diagrams here, you can more or less, you can do these if you'd like, just to help you understand how many electrons are in each orbital, but you have to realize that spatially these are incorrect, right? It's not 2-dimensional. So if you wanted to do hydrogen, then you would just put one little circle in that first ring around the nucleus, representing 1 electron in the outermost shell. And then you can do the same for carbon, oxygen, and nitrogen if you'd like. Water. So using the red and yellow balls are the hydrogens and oxygens. Which end of the water molecule has a slight negative charge? Again, you can look at the pictures I sent you. You can figure that out. It's pretty easy. What type of bonds hold the oxygen and hydrogen atoms together? The type of bonds that hold them together are polar covalent bonds, again, because the electrons are not equally shared between the 2 atoms. Okay, and then if you were to arrange those water molecules on your bench to show how the one water molecule binds to another water molecule, now we're talking about hydrogen bonds, right? So the hydrogen of one water molecule would form a hydrogen bond with the oxygen of the next water molecule. Okay. Okay, now moving on to glucose, a very important macromolecule. All you need to do here to answer these questions is basically count how many carbons there are. That's pretty easy to do. Just cross them off as you count them. The chemical formula, again, make sure you use subscripts when you're writing formulas. So you would count up the carbons, and then the subscript with how many carbons, count up the hydrogens, subscript with how many hydrogens, count up the oxygens, subscript with how many oxygens. What type of bond holds the atoms together in a molecule of glucose? Those would be polar those would be non-polar covalent bonds. Electrons are shared, but there's no net charge on the glucose molecule. The human body, what polysaccharide can be formed when individual molecules of glucose are bound together? When your blood glucose level goes up and you have excess glucose, your liver will actually convert that excess glucose into a storage form of carbohydrate, which is called glycogen. The glycogen is then stored in the liver. When your blood glucose levels go down, the liver will convert the glycogen back to glucose and release it to the blood. Lipids. Lipids, these are fatty acid chains. Again, long chains of hydrogens and carbons. And they form two different types of lipids, which if you go back to the beginning of the lab, it tells you what two types of lipids those are and what those are primarily used for. And what we want you to do here, because you can't actually model these, is just draw the two types of lipids they're telling you here. Okay, just draw them out on your paper. And then you'll be able to answer those questions. The next macromolecule it talks about here are proteins. Proteins are made of little units called amino acids. And an amino acid consists of, and there's a picture of it right here for you, a central carbon with an amine group, which is the NH2. You can think of proteins like a string of pearls, or like little pop beads. When you're a kid, you played with little pop beads and you made little chains of beads. The amino acids are all arranged like that, like beads in a chain. And how the sequence of those amino acids is imperative to determining the function of that protein, along with how it's folded up and bound to itself. But the sequence of amino acids determines the function of the protein. For example, you could have the word one, O-N-E. I could rearrange those letters to write neon, to write neo, N-E-O. Right? The same, if you will, amino acids arranged differently, two very different functions for those words or for those proteins. Okay? In cells, there are also two basic types of reactions that happen. And again, I think there's a picture of this in the review sheet that I gave you. Yep, right there. There are either dehydration synthesis reactions or hydrolysis reactions. So we know dehydrate means to lose water, right? Synthesize means to build. So dehydration synthesis reactions, we're going to pull a hydrogen ion off of one molecule and a hydroxide from another. Those are going to form water, and we're going to hook those two molecules together. So we're building something by removing water. Hydrolysis is the opposite reaction. We're going to take water, we're going to put hydrogen ions on one molecule, hydroxide on the other, and it splits the macromolecule apart. So hydrolysis is the type of reaction used by your cells to break things down. Okay, so that should help you with that part of the lab. Okay, and that in a nutshell is the first lab, the first part of the lab for lab 3. I am going to stop this now and I'm going to record another video. It's easier to upload them if they're shorter, and I will upload both of them and send them off to you. So I'm going to stop this one now and I'm going to pick up with pH, which is the second part of the lab.